Hello everyone, we are Team 3. My name is Vanessa and Skylar and Amy are my teammates, and today we'll be talking to you about immunohistochemistry. So immunohistochemistry, or IHC, is an immunostaining technique that is able to visualize and document the distribution and localization of specific cellular components within tissue samples. IHC is commonly used in clinical and research laboratories and has a variety of applications including disease diagnosis, drug development, and biological research. The concept behind IHC is that it uses antibodies that are able to specifically bind to antigens in a biological tissue sample, where the binding of the antibody-antigen complex can be visualized using an enzyme that is linked to a specific antibody. Common enzymes that are used are horseradish peroxidase, which produces a brown stain, and alkaline phosphatase, which produces a blue or purple stain. Since IHC uses enzymes rather than a fluorescent dye, you are able to store your samples for a longer period of time and reduce autofluorescence. So, the way that IHC works is that you will have a fixed tissue sample that will have an antigen that the primary antibody is able to bind to. Then a secondary antibody that is linked to your enzyme will bind to the Fc region of the primary antibody. When this binding occurs, your enzyme will react with a colorless substrate and will produce a colored product that can be viewed via light microscope. When using the IHC method, there are some important parameters to take into account to ensure you generate good results. First, you'll want to make sure that you are properly mounting your tissue samples in order to avoid nonspecific binding. Non-frozen tissue samples need to be fixed gently using chemical fixation, and frozen tissue samples are fixed after the antibody reaction has occurred in order to make sure that the antigen will be recognized by the antibody. Second, you will need to make sure that you are choosing the correct antibodies for your tissue sample. The secondary antibody must be raised against the IgG of the animal species in which the primary antibody has been raised. So, for example, if you have a human tissue sample, you can have something like a mouse anti-human IgG, and then you can have a secondary antibody that is linked to the enzyme that is a goat anti-mouse IgG.
The results from immunohistochemistry are insoluble colored products or precipitations. They are localized at the site where they are formed, which can be observed directly under a light microscope. They provide information on expression of antigen on tissue, protein localization, cell morphology, cell division, and distribution of protein. The common way to analyze the data from immunohistochemistry is to measure the staining intensity, which can be measured by optical density and pixel analysis software. Moreover, semi-quantitative scoring system is also commonly used. On the right side are two pictures of stained human cells. The top one is stained by horseradish peroxidase. The bottom one is stained by alkaline phosphatase.
In clinical practice, immunohistochemistry is often used for cancer diagnostics. By using specific known tumor markers as the antigens, physicians can use IHC to diagnose tumors as benign or malignant, determine the stage and grade of a tumor, and identify cell type and origin of metastasis in order to find the primary site of a tumor. The image on the right depicts an example of using IHC to detect P21, a cyclin-dependent kinase inhibitor, in human colon carcinoma tissue. P21 plays a role in regulating the cell cycle, and since P21 expression is lost in most colorectal cancers, the visualization of P21 localization and intensity can help physicians to determine a cancer diagnosis. The process of using primary antibodies to detect antigens and then secondary enzyme-conjugated antibodies that will catalyze in the presence of substrate and chromogen is only possible due to immunoengineering. The development and continued improvement of conjugated antibodies through immunoengineering has allowed for these enzyme labels, which are detectable through a light microscope.